Good day, everyone. Today, me, Abhilasha, along with my group members Natasha, Rachel, Siddharth, and Omar, will be presenting on the databases and tools for breast cancer gene analysis.
Today I'll be introducing the role of bioinformatics in modern biomedical and drug discovery research. Bioinformatics is the fusion of computational and biological sciences. This integration has significantly transformed how we approach medical research, particularly in drug development, disease diagnostic, and personalized medicine. As highlighted in COP in 2020, the transformation is driven by our growing ability to analyze biological data through computational tools. With the rapid rise of genomic and proteomic data, manual analysis has become nearly impossible. Instead, researchers now rely on advanced bioinformatic tools not just to store this vast information but also to process it and uncover meaningful patterns within complex datasets. Morakit and colleagues in 2021 emphasized that this shift is essential to manage the scale and complexity of current biological research. In drug discovery, bioinformatics allows us to identify potential drug targets and conduct virtual screening of compound libraries through in silico simulations. These methods are highly efficient, enabling researchers to predict pharmacokinetics and toxicity profiles before clinical trials even begin. According to Lee et al. In 2021, this significantly reduces the time and cost involved in traditional drug development pipelines. Overall, bioinformatics is no longer just a supporting tool, it has become a core driver of innovation in biomedical research and personalized treatment strategies.
To detect genetic predisposition to familial breast cancer, a structured bioinformatics workflow is applied after sequencing. It starts with DNA sequencing, typically using whole exome sequencing WES, or targeted gene panels that include high risk genes like BRCA1, BRCA2, and TP53. These methods focus on the most relevant coding regions. Next is quality control, where tools like FastQC filter out low quality data and correct sequencing errors to avoid downstream bias. Then, sequence alignment is performed using tools like the BWA, which maps the clean reads to a reference genome such as GRCH38, generating BAM files that show how the DNA sequences align. After that comes variant calling, using tools like GATK or Freebase to identify sequence changes, like SNPs and small insertion or deletions, which is recorded in a BCF file. These variants are then fed into annotation tools such as ANNOVAR, SNP-EFF, or Ensemble VEP, which provides crucial biological context such as how a mutation might affect protein function or clinical outcomes. Next, pathogenicity prediction tools like PolyFan2, SIFT, and Mutation Tester assess whether these changes are likely to cause disease. To confirm clinical relevance, these variants are cross-referenced with databases like ClinVar, BRCA Exchange, and BBSNP to see if they've been reported before and what the new impacts are. Finally, these variants are classified based on ACMG guidelines such as pathogenic, likely pathogenic, benign, or VUS, meaning variant of uncertain significance. This classification informs diagnosis, risk evaluation, treatment, and genetic counseling. Overall, this pipeline ensures both high accuracy and clinical relevance in identifying breast cancer risk.
Hi, I'm Omar Aso. I will talk about the role of bioinformatic tools in familial breast cancer. Bioinformatics tools are essential in identifying genetic mutation linked to inherited breast cancer, especially in high-risk genes like BRCA1 and BRCA2. They help process large amount of sequencing data, highlight diseases, cause variant, and translate this information into something useful for clinical discussion making. This support personalized medicine, allowing doctors to offer better screening, treatment plans, and family counseling based on person's genetic profile. On the right, you can see a simplified workflow starting from sequencing to clinical discussion making. Each step involves specialized tools that I will explain next. This slide shows the main tools used in each steps of the bioinformatics workflow. For quality control, we use tools like FastQC and Tromantic to clean the sequencing data. Then BWA and some tools help to align the reads to the reference genome. GATK and FreeBias are used for detecting genetic variants like SNPs and ANDLs. Once we find them, annotation tools like ANNOVAR and SnapEFF add information about how those mutations affect gene and proteins. To predict if a mutation is harmful, we use tools like SIFT and polyphen2 and mutation tester. Finally, we compare these variants against clinical databases like ClinVR and BRCA exchange to check if they are already known to cause diseases. All of these tools together help doctors understand genetic risks and make informed discussion about treatment or prevention.
I'm going to talk about how certain tools are used in clinics to deal with inherited breast cancer. First is phenolphine 2. This tool helps predict if a BRCA variant is harmful or harmless, especially when the mutation is new and not in databases. Doctors and labs can use it to decide if a mutation could increase cancer risk. Next is genome AD. It shows how common or rare a variant is in the general population. If it's common, it's usually harmless, but if it's rare, it could be linked to a disease. This helps doctors focus on the variant that matters most. Then we have ClinkVar. It's used by labs like Moderate Genetics and Inval to classify BRCA1 and BRCA2. This supports real-life decisions like whether a person should get genetic counseling or even preventive surgery. Lastly, BioAD ICEA, which is used through the CanRisk platform, is used by the UK NHS to estimate a woman's risk of developing breast cancer. Based on the results, doctors can recommend early screening, genetic testing, or preventive medicine. In short, these tools can help turn complex genetic DNA into real action, improving early detection, prevention, and personalized care.
Next would be the case study and examples in database and tools used for gene analysis in breast cancer. The first one would be whole exome sequencing, which is WES. WES has been used to identify variants in high risk families. This has been used in a study by Akbari in 2021. Databases such as CleanVar database has also been used for cross-references variants with clinical data. As for computational and prediction tools, SIFT and polyphen 2 has used to be assess the functional impact of amino acid changes. This has been used by Ng and Hanikoff in their study in 2003 and by Adube in 2010. An example in population and frequency filtering is the database like 1000 Genomes and GenomeAd that was used to help remove common variants in breast cancer gene analysis. For expanded gene analysis, a study by Couch in 2017 found additional susceptibility genes using the data from TCGA and also BIC. As for bioinformatic platforms, platforms like Galaxy streamline processes such as sequence alignment, variant calling, and also annotation in breast cancer gene analysis.
In conclusion, bioinformatics is a game changer when it comes to detecting breast cancer gene, making it faster, early, and far more accurate. Thanks to high-speed sequencing and smart prediction tools, we can now spot genetic variants with more confidence. This means better diagnosis, clearer risk assessments, more personalized treatments, and well-informed genetic counseling. It also helps guide decisions for future pregnancies and long-term plans. In short, bioinformatics empowers patients with the right tools and knowledge, and as this technology improves, so will the outlook for those with hereditary breast cancer risk. Thank you.